Hello, this is Mr. Chambers, welcoming you to the Beaver Science YouTube channel. In this video, we will be taking a look at one possible explanation of the origins of life first proposed by Oparin and Haldane independently in the 1920s. This has been explained as the chemical evolution process, and later this explanation was tested by a couple of scientists at the University of Chicago by the name of Harold Urey and Stanley Miller. This graphic, or this figure, actually kind of illustrates Miller's experiment, and we're going to draw this up, and I'm going to try to give you a brief explanation of how this really was designed to test Oparin and Haldane's thoughts or ideas about life originating through chemical processes. And if you hear that chemical evolution, basically that refers to life originating from organic molecules in the presence of energy over long periods of time. The notes that go along with this come from the second sequence of PowerPoint notes, which are from chapter 24 in your textbook over early life and the diversification of prokaryotes. So we'll start by basically getting a blank sheet of paper, either notebook paper or typing paper, and we'll orient this in landscape. It's going to be a fairly busy drawing, and it's going to take us a little while, and so settle in and hopefully we can do this in one take. At the top of this, of your paper, just simply label this Miller's experiment. And I'm going to start over here kind of a little bit off center, more towards the left-hand side of the paper, as we're going to work a little bit more to the right as we progress through the drawing. So Miller's experiment, this was done in 1953. So nearly 70 years ago today at the University of Chicago. And Stanley Miller was fairly young at the time when he did this. But what Miller was trying to do basically was to replicate the Earth's reducing atmosphere. And you'll see that term, reducing atmosphere. And reducing atmosphere can be described in a couple of different ways. One is based on what molecules we think were in the Earth's atmosphere at 4.5, 3.5 billion years ago, which we think it was primarily a hydrogen-based atmosphere with methane, ammonia, carbon dioxide, carbon monoxide. But you can also describe a reducing atmosphere based on what's not there. And we think that there was very little or no free oxygen in the Earth's kind of primitive atmosphere. So one of the first things that Miller needed basically was a heat source. And so the Earth would have been very hot at this period of time. It's getting bombarded with cometary impacts and a lot of energy. And so we need a heat source. The Earth would have been above 100 degrees Celsius at this time. And so any water vapor or any water on the surface of the planet would have been in the vapor form, not the liquid form. And so Miller didn't have one thing at his disposal, which we don't have today as well, which was a time machine. And so we can't go back 3.5 billion years and actually see these processes of chemical evolution. So what he did was, instead of going back and observing it, he made a model or an analogy for the Earth's reducing atmosphere. Basically, he built this closed loop or this closed system primarily out of glass, and tried to replicate the Earth's reducing atmosphere to see if it was possible for life, or at least the precursor molecules to life, organic molecules, to be created under these conditions. So he started with a Bunsen burner or heat source. And then he needed what we know all living organisms today need, which is water. And so he heated that water up in a closed vessel, in this glass vessel, over 100 degrees Celsius, and he pushed that water from the liquid form to the vapor form through this series of glass tubes. So as the water heated up, it went up to above 100 degrees Celsius, and it kind of went through this phase change, and it went from the liquid form to the vapor form. You can kind of hear my pen skating across there. So once the water hit that 100 degrees Celsius mark, again it went from that transition to liquid water to vapor. As he pushed this water vapor through this closed system, he built another vessel where the water came in and exited. And this essentially became Miller's reducing atmosphere. So what he put in this portion of the model were the gases that we think were in the Earth's primitive atmosphere, which was methane, among others, ammonia, and then a very important one, hydrogen. Okay, so essentially this is Miller's analogy of the reducing atmosphere. He also needed an energy source. And so the energy source that Miller introduced was an electrical spark. And that electrical spark really is meant to be an analogy to lightning, one possible energy source that could have helped jumpstart these chemical reactions in the Earth's primitive atmosphere. So this is his reducing atmosphere, essentially. With an energy source. And so then he circulated that water past those gases in the presence of an energy source, and he pushed it back out the other side. So as that water moves through the Earth's reducing atmosphere, it's going to continue on through this loop. And down here, he added something to move the water back from the liquid form to the vapor form. And this something was what he called a condenser. So we're going to condense the water from the vapor form back to the liquid form. And so he surrounded this glass tube basically with this condenser, and what he did was he circulated cold water around this tube that's coming out of his reducing atmosphere. And as that vapor moved through this tube, that cold water cooled that water vapor and it went back to the liquid form. And then he just kept it in a loop. And so he brings it back down and around here. But now that water is in the liquid form. But he doesn't push it all the way back to the original vessel just yet. He builds in a little valve because he's interested in collecting a sample of this water that has been circulated through the reducing atmosphere. So he puts a little valve in here that he can open and close. And so now this liquid water moves past this valve, and down here he has the old Erlenmeyer flask. And the Erlenmeyer flask is where he's going to collect his sample. And so as this water moves through and past this valve, he opens the valve and he collects a sample for analysis in the Erlenmeyer flask. So this becomes, and the rest of the water, liquid water, just continues on back to this vessel, and it becomes kind of this closed loop that essentially is an analogy of the Earth's reducing atmosphere. And kind of remember what we're talking about here and what we're trying to understand is, is it possible for organic molecules or maybe even life to eventually originate under these types of conditions? And so this is his sample. For analysis. And he takes this sample and he runs a chemical analysis on this sample. And lo and behold, he doesn't find living organisms. And remember, there's a couple of things that Miller doesn't have. One of the things that he doesn't have is a time machine, so he can't go back and watch or see this process under these types of conditions, which we think were occurring at 3.5 billion years ago. He doesn't have a time machine. The other thing is he doesn't have 3.5 billion years. And so he doesn't have that long periods of time in order to be able to run this to see if it's possible to go ahead and move from really just simple organic molecules to macromolecules to protocells, kind of the precursors to the first living cells, and then self-replicating molecules, the nucleic acids, RNA and DNA, and eventually step over and kind of cross that magical line where we go from organic molecules to the first living organisms, which we see fossil records to at about 3.5 billion years. But what he did find in this sample were organic molecules. And if you see that term organic in biology, that means that we're talking about molecules that have a carbon atom associated with them. And the organic molecules that he found were not these macromolecules, but these simple molecules that are kind of the building blocks for the macromolecules that we know are common in all living systems. What he found were, and I'm just going to abbreviate it here because I'm a little bit short on room, were the AA, amino acids. And there are 20 naturally occurring amino acids in living systems. But amino acids, if you start to combine these amino acids, they form one of those important, really important macromolecules that we find in cells that are kind of the structural and functional unit of cells, which are proteins. And living things, cells, are littered with proteins. Proteins are both structural and functional, and so they're what the cell uses to make components, ribosomes, globular proteins in the membranes, all of these structural components of the cells. Proteins are an important component of that, but they're also functional proteins, and functional proteins act as enzymes that speed up or catalyze chemical reactions. Okay, so we get kind of maybe the first step to living organisms, which are these simple organic molecules, these amino acids. If you picture these amino acids kind of combining and forming these proteins, a little bit more complex organic molecule, and under laboratory conditions we can get these amino acids and eventually proteins. And we can also get some important organic molecules that we know are essential to life under these types of conditions, basically in the lab. And when we generate another important type of molecule, macromolecule to living organisms called lipids or fats, those lipids will spontaneously form a membrane and surround these proteins. Okay, now this is not a cell, but this structure, which your book will refer to as a vesicle, is kind of an important step towards, and has many of the characteristics that cells demonstrate today. And so this structure represents a combination of amino acids. And so those are our proteins. And then surrounding these proteins is a lipid membrane. Now, Miller's experiment has since been replicated, and they've tweaked it a little bit. And, you know, this idea or this thought of organic molecules, simple organic molecules, and then macromolecules, and eventually vesicles, which is how your book will describe these membrane-bound proteins. And then these vesicles becoming a little bit more complex and forming what are called protocells. And eventually, protocells are not considered living, and so the protocells are abiotic. And so we're kind of looking at this possible explanation for life evolving under, you know, these primitive conditions in a reducing atmosphere. Protocells start to demonstrate some of the properties of living organisms. You know, you have a membrane, you have proteins, but they can't do all the basic characteristics of life. And so they may not necessarily be capable of acquiring nutrients or reproducing or responding to stimuli. But eventually, step by step, we're moving towards those first living organisms that we see fossil records of at about 3.5 billion years. And those protocells at some point kind of cross over this, you know, magical barrier where they become the first living organisms on the planet and become established as the first true cells on the planet, which are the prokaryotes. And we still have prokaryotes with us today, and the prokaryotes have been around a lot longer than we have, and probably will be around a lot longer after we're gone. If you see that term prokaryotes, pro means pre or before, and karyote means nucleus. So these are organisms that don't have a true membrane-bound nucleus. And one of the terms that you want to associate with prokaryotes is bacteria. So we think the first living cells to originate show up in the fossil records at about 3.5 billion years. This is one possible explanation of how we might have gone down this pathway. Your book will actually talk about 4 stages, and I'm going to just jot those down really quickly up here and kind of wrap this up. But refer back to your notes and your textbook if you have it, and hopefully this will help provide you with a little bit more structured explanation of those notes. So, 4 stages that could have led to simple cells as proposed by your book. And if proteins are the structural and functional unit of cells, cells are the structural and functional unit of life. And so we know that all living organisms, from the cell theory, Schleiden and Swan in the 1850s, from the cell theory, all living things are made up of cells. Cells are the structural and functional unit of life. And later, Virchow comes along with the third tenet, but those are the 2 important ones to think about here. So the first step is we get small organic molecules. And for example, amino acids, as we see created through this process at Miller, where he replicated the Earth's reducing atmosphere. From those small organic molecules, we get the larger molecules, which are the macromolecules that we know that are found in all living organisms. And those 4 macromolecules are the proteins, the lipids, the carbohydrates, and then lastly, the nucleic acids. Along the way, after we get the macromolecules, we get what we think are the precursors to the first living cells, the protocells, and then the last kind of piece of the puzzle are the self-replicating molecules, which are the nucleic acids, RNA, and then eventually DNA. Okay, I'm gonna show you another copy of this that might just be a little bit cleaner, but I'm gonna try to get this centered and give you just 1 minute so you can see another version of this. I think it's pretty close to the same thing, but I'll see if I can kind of zoom out and get this centered where you can read, hopefully, all of the information and get a little bit better look at this. So I know this is a lot, and we kind of covered a lot of ground here, but hopefully this will help you to visualize and have a little bit better understanding of one possible explanation, chemical evolution processes that could have led us down to the pathway to the first living organisms on the planet at more than 3.5 billion years ago. So thanks for joining, and come back and see us again here on Beaver Science.